Hi everyone! In this lecture we'll finish our coverage of plant growth and development with a look at secondary growth, the development of roots, and a look at some environmental factors that affect growth. Shoot apical meristems are responsible for growth in height or length. This type of growth is called primary growth. However, in order to grow tall, plants must also be able to grow in diameter in order to support their own weight and to supply sufficient water needed for photosynthesis. An increase in a plant's diameter is referred to as secondary growth. Secondary growth results from a new type of meristem, lateral meristems. Lateral meristems are similar to shoot apical meristems in their ability to produce new cells that grow and differentiate, but they also differ in several ways. Lateral meristems are not found at the tip of the stem, but rather form cylinders of cells that surround and extend the entire length of the stem. Lateral meristems produce cells that grow in diameter, but not in length, because these meristems form after elongation is complete. Lastly, lateral meristems become larger over time because as the stem becomes thicker, as their circumference increases, the number of meristem cells needed to encircle the stem also increases. There are two lateral meristems, the vascular cambium and the Cork cambium, that together they drive secondary growth. Vascular cambium is the source of new xylem and phloem, called secondary xylem and secondary phloem. The vascular cambium is responsible for most of the increase in the stem's diameter. The secondary xylem makes up what we would call wood, and plants with secondary growth are often referred to as woody. As plants with secondary growth increase their size, the epidermis, which was formed during primary growth, ruptures. The second lateral meristem, called the Cork cambium, functions to renew and maintain a protective outer layer. The vascular cambium produces new cells on both of its sides, towards the inside of the stem and towards the outside of the stem. The cells on the inside differentiate to become the secondary xylem, and the cells on the outside become the secondary phloem. All tissues that are produced to the outside of the vascular cambium are collectively called the bark, and this includes the secondary phloem and the Cork cambium, which we'll discuss in a moment. As the vascular cambium forms new secondary xylem cells along its inner face, the newly formed cells push the vascular cambium outward. The vascular cambium accommodates this outward movement by adding new cells that increase its circumference. The secondary phloem is unable to form additional cells. As the production of secondary xylem pushes the vascular cambium outward, the phloem becomes stretched and damaged, so new secondary phloem must be continually produced. The secondary xylem can be divided into heartwood and sapwood. Heartwood makes up the innermost part of the stem and is often darker than the sapwood due to the presence of some chemicals like resin. The xylem found within the heartwood is not able to transport water. As the xylem ages, it loses its ability to transport water, leaving only the xylem close to the vascular cambium functional. This functional xylem is found in the sapwood, which rings the heartwood. Seasonal climates and environmental conditions can impact the growth of the secondary xylem. These impacts can be seen in the stem's growth rings. In the spring, the vascular cambium produces large cells with thin walls. This is the light part of the growth ring. As growth slows down in summer, the cambium produces smaller cells with thicker walls, and this is the dark part of the growth ring. This alternation over the growing season makes it possible to determine a tree's age by counting the rings. The width of each growth ring also indicates how much wood was laid down that year, which reflects the environmental conditions experienced that year. When there is adequate moisture and permissive temperatures for photosynthesis, more wood and a wider growth ring is produced relative to years of drought. Not all trees develop growth rings, however. Trees that are found in regions that do not experience seasonality in rainfall or temperature grow at a continual rate throughout the year. This means that there are no visible growth rings produced. On this slide is a picture taken by Dr. Bauma in the Monteverde Cloud Forest Reserve in Costa Rica, and it shows an example of such a tree. For a stem that is actively increasing in diameter, the Cork cambium is the lateral meristem that maintains a protective outer layer around the stem. The cork cambium initially forms from cortex cells that regress to their earlier state as meristem cells. Over time, as the cork cambium actively divides, it becomes increasingly distant until it is cut off from the phloem, which is the source of its carbohydrates. When it becomes sufficiently cut off, a new cork cambium forms within the secondary phloem. Each cork cambium is relatively short-lived, and in many cases it forms in patches rather than in a continuous layer. This is why bark oftentimes looks patchy and fissured, like in this ponderosa pine to the left. Now in this cross-section of bark from an English walnut, you can see these lines here. These are the cork cambium, and they surround larger patches of cork. This layer down here is the secondary phloem. Cork refers to cells produced by the cork cambium. As cork matures, the cells become coated with suberin. Suberin is a waxy compound that protects against mechanical damage and serves as a barrier against the entry of pathogens and the loss of water. The flip side of the protective nature of suberin means that this layer of waxy non-living cells also impedes the inward diffusion of oxygen, which is needed to meet the respiratory demands of living cells. To combat this, the cells of the outer bark are less tightly packed in regions called lenticels, and these lenticels allow oxygen to diffuse into the stem. The secondary xylem fulfills two roles as the tree grows. It provides the stem with strength and stability, allowing the tree to grow tall and support a large crown. And, like the primary xylem, the secondary xylem transports water and nutrients from the roots to the leaves. A single cell type fulfills both of these functions in gymnosperms, like pines, seen in this photo to the left. In these plants, cells on the xylem side of the vascular cambium differentiate to form tracheids. Tracheids are single-celled xylem conduits that have thick lignified walls and are non-living and hollow at maturity. In angiosperms, on the other hand, the cells in secondary xylem mature into distinct types that separate the functions of water conduction and mechanical support. An example of angiosperm wood can be seen in this photo to the right, which shows a section through a Douglas fir stem. As an aside, the Douglas fir is actually the state tree of Oregon. Support for the growing plant is provided by fibers. Fibers are narrow cells with extremely thick walls and almost no lumen or interior space. Vessel elements are wide cells that function in the transport of water. Angiosperm wood is more efficient at water transport than gymnosperms because these vessel elements form multicellular xylem vessels with open connections between them. Because of this, angiosperms have higher rates of water flow through their stems compared to wood that contains only tracheids. Greater water flow enables angiosperms to sustain higher rates of carbon dioxide uptake and therefore higher growth rates. Roots function to anchor plants in the ground while permitting vascular plants to draw water and nutrients from the soil. Roots allowed vascular plants to expand their habitats from extremely wet environments to drier ones and to provide the support needed for plants to grow taller. Before we make the transition to discuss root structure, we'll be contrasting stem growth and root growth in this summary table. Roots grow in the same way as shoots, with new cells produced at their tip. The root apical meristem is the source of new cells. After cell division, these cells undergo elongation, then complete the differentiation only after elongation has ceased. There are regions of cell division, elongation, and maturation in roots, just as in stems. However, roots differ from stems in several ways. Firstly, roots are much thinner than stems. Their thinness optimizes their surface area to volume ratio, and as a result, increases their uptake of water and nutrients. Their thinness also allows roots to bend around soil particles and make use of channels created by invertebrate animals that burrow through soil. Secondly, the root apical meristem is covered by a root cap that protects it as it grows. The shoot apical meristem is not covered. The root cap can be rubbed off or damaged during growth in the soil, so it is continually being supplied with new cells produced by the root apical meristem. Another difference is that the root apical meristem does not produce any lateral organs, whereas the shoot apical meristem produces leaves. both roots and stems are capable of primary and secondary growth, though they do differ in their branching patterns. Roots typically have more extensive branching than do stems. This longitudinal section shows the regions of a root. Starting at the tip, we have the zone of cell division, where we find the root apical meristem, where new cells are produced, and the root cap that protects it during growth. As we move up the root, there is the zone of elongation, where the root cells increase in length, followed by the zone of maturation. In the zone of maturation, the cells have stopped growing and will finish differentiation. Root hairs are also found in this region. This cross-section shows the concentric tissue layers of a typical root. We have the epidermis on the outside, and as we move inwards, this is followed by the root cortex, the endodermis, the pericycle, and then the vascular cylinder or bundle. As you may remember from a previous lecture, the endodermis plays an important role in the selective uptake of nutrients. Encircling each endodermal cell is a waxy band called the Casparian strip. This strip forces all the material that is destined for the xylem to enter the endodermal cells and pass through their plasma membrane, rather than travel through the endodermal cell walls. The vascular bundle or cylinder is found at the center of the root. The distribution of the xylem and phloem tissue varies some between plants, but this cross-section displays a pattern that is common to many. The xylem tissue runs through the root as a fluted cylinder, so that it appears as a star in cross-section. The number of points to the star varies from species to species, but the one here has 4. In the grooves of the star is the phloem tissue. Just like with the stem, the xylem tissue is closer to the interior, and the phloem tissue is closer to the exterior of the root. The hormone auxin plays a central role in differentiating which cells will become xylem and phloem in the root. The phloem transports auxin produced by shoot meristems and leaves downward through the mature stem and into the roots. When the auxin reaches the end of the fully formed and mature phloem in the roots, it is moved by polar transport from cell to cell until it reaches the root apical meristem and the root cap. Movement of auxin through the zone of elongation leads to the formation of procambial cells. These cambial cells subsequently differentiate into phloem and xylem. This transport and movement of auxin through the stem and the developing root ensures and establishes the vascular continuity between roots and leaves. As I mentioned on the previous slide, roots can also undergo secondary growth, thus increasing their diameter and adding new xylem and phloem. The vascular cambium forms at the interface between the xylem and the phloem as it does in stems. To ensure that the connection between the secondary vascular tissue of the stem and the roots is unbroken, the vascular cambium must be continuous from the shoot to the roots. In older roots, a cork cambium is also formed to maintain the outer protective covering for the roots. I pointed out the pericycle briefly on the previous slide. This is the single layer of cells found just inside the endodermis and next to the vascular bundle. Here we'll look at it in a little more detail and the important role it plays in the roots. Roots branch quite a bit, particularly when compared to stems. The extensive branching of the roots is necessary to create enough root surface area to supply the water and nutrient needs of the leaves. A new root apical meristem must be formed in order for a root to branch and form a lateral root. These new root meristems, and subsequently the new roots, develop from the pericycle. As the pericycle is next to the vascular bundle, the developing new root is connected to the vascular system from its beginning. Because the pericycle is located in the interior of the root, the new root meristem and root must grow through the endodermis, cortex, and epidermis before reaching the soil. These two lower pictures are images that I took of a cross-section through a corn root that is forming a new lateral root. The layout of the vascular tissue is a little different in corn, but the new root still originates from the pericycle. By initiating new growth from the pericycle, new roots can form anywhere along the root. As a result, root systems have tremendous flexibility, and roots can proliferate in local regions where nutrients are abundant. The majority of new roots die and are shed after only a short period of time. The continuous growth of new roots represents a major expenditure of carbon and energy for the plant, but it also allows the root system to respond to changes in the availability of water and nutrients. New roots can form from existing roots and from stems. The structures and functions of roots are diverse. How a plant's roots are distributed in the soil has a tremendous impact on the ability of the root system to supply the shoot with water. Some plants produce deep roots that provide access to groundwater independent of rainfall. Some plants produce shallow spreading roots to capture as much water as possible from rain showers. Some plants produce roots that allow them to adhere to its surrounding environment. So for example, here we have some poison ivy climbing a tree trunk. Plants with prop roots are able to grow tall despite their slender stems. Here we have a walking palm that has prop roots that serve to support it. Swollen roots are produced in some plants to store resources such as starch. Here's an example of that in yams. And then breathing roots are produced in plants that grow in water, like these black mangroves, to provide an easier pathway for oxygen to diffuse into the roots in plants that grow in water. We're going to switch gears a little bit and discuss ways in which plants are able to modify their growth and make use of their capacity for growth throughout their lifespan. Plants are able to modify their growth in response to changes in their environment. They can sense the world around them in three principal ways. First, they have photoreceptors, which provide information about the availability of light needed to drive photosynthesis. And we have covered photoreceptors a little bit previously. Plants also have mechanical receptors. These receptors provide information about physical influences from their environment, such as wind or gravity. And lastly, there are chemical receptors, which provide information about the presence of specific chemicals and chemical gradients. Stems and roots control their orientation by environmental cues, which make use of these different receptors. The bending or turning of an organism in response to an external signal, such as light or gravity, is called tropism. Movement towards the stimulus is a positive tropism, and movement away is a negative tropism. Because plant stems will bend towards light, they are positively phototropic. Plant stems are also negatively gravitropic because they grow upwards against the force of gravity. Plant roots, on the other hand, are positively gravitropic and negatively phototropic because they grow down with the force of gravity and away from the light. As we'll see in the next few slides, plant hormones play an important role in the interpretation and transmission of the information gained by the receptors and turning it into an appropriate response. In 1926, a Dutch graduate student, Frits Wendt, sought to understand how the perception of light by the tip of a plant led to a growth response further down the stem. The reasoning behind his experiments was that if a chemical was the signal from the shoot tip to the stem, then he could isolate the chemical by cutting off the tip of a growing plant and placing the tip on a small block of gelatin. As the chemical moved out of the shoot tip, the gelatin block would capture it like a trap. In his first experiments, he removed the plant tip from an oat plant and placed it on gelatin for 1 to 4 hours. During this time, the plant ceased to grow. Wendt then placed the gelatin block on top of the cut plant and observed that growth resumed. In his second experiment, Wendt placed the gelatin block to one side of the top of the cut plant. Growth resumed such that the plant bent away from the side with the gelatin block. From his experiment, he concluded that a signal which stimulates growth was produced in the tip of the plant, and that it travels downward through the stem towards the roots. This signal is now known to be the plant hormone auxin. Plants produce photoreceptors that allow them to detect and respond to light. In the case of phototropism, it is photoreceptors that respond to blue light that trigger the phototropic response. As light shines on a plant, only the photoreceptors that are exposed to the light activate. This activation is thought to result in the movement of pen proteins towards the shaded side of the stem. Recall that pen proteins are required to export auxin from cells. The concentration of pen proteins on one side of the cells results in auxin to be transported preferentially to the shaded side of the stem. The outcome is a high concentration of auxin on the shaded side of the stem and lower auxin levels on the illuminated side. In stems, the presence of auxin stimulates cell expansion. This means that the stem will grow faster on the shaded side than it does on the illuminated side, causing the plant to bend towards the light. In roots, phototropism is similar in that illuminating a root from the side causes auxin to be redistributed towards the shaded side of the organ. With roots, oxygen is transported to the shaded side of the root cap and then moves up the root into the zone of cell elongation. Roots differ from stems in that higher oxygen concentrations decrease the rate of cell elongation, thus causing roots to bend away from the light. The reason that oxygen redistribution leads to a different outcome in roots compared to shoots is that in roots, high concentrations of oxygen trigger the production of the hormone ethylene, which reduces cell elongation. Once a seed has germinated, the newly germinated seedling must reach the soil surface before its resources stored in the endosperm or cotyledons are used up, or it will die. The seedlings from seeds that were germinated in the dark are very pale with thin, highly elongated internodes and suppressed leaf development. This is because they're putting all of their resources into elongation in order to increase their chances of reaching the light. We can see an example of that here. We have long internodes, and it is a very pale green compared to its sibling that was germinated in the light. If the seedlings do reach the light, internodal elongation slows, and green leaves expand to capture energy from the light. These changes in growth patterns are triggered by several photoreceptors that detect and signal the presence of light. One photoreceptor absorbs blue wavelengths of light, and another, the phytochrome, responds to red and far red wavelengths. When plants detect mostly far red light, which indicates they're in the shade, some plants respond to the presence of shading plants by elongating more rapidly and suppressing branching, which allows them to maximize height growth and make it more likely that they're going to reach full sun, and that's an example here. You may remember this figure from a previous lecture, where we discussed the impact the shading of plants had on seed germination. Recall that as sunlight passes through leaves, the red wavelengths of light will be absorbed by chlorophyll, but the far red wavelengths are not. This is why in the shade, the plants will detect far red light and thus the presence of overhead plants. Growth in plants also responds to gravity. If we focus on roots for a moment, starch-filled organelles called statoliths are thought to provide roots with information regarding their orientation relative to gravity. Starch is a large molecule whose density is greater than that of water. This means that in a cell, statoliths will sink to the bottom of the cell. In the root caps, the statoliths press on the cytoskeleton or on membranes, and so provide information about gravity to the plant. In other words, they let the plant know which way is down. This is then thought to trigger the movement of oxygen towards the lower side of the root. In stems, similar organelles in bundle sheath cells are thought to play a similar role. To talk about water's influence on roots, we're returning to our table of the major hormones because we need to talk about ethylene and abscisic acid. During drought conditions, plants will produce more roots with fewer branches, and these roots will penetrate deeper into the soil to increase the chance of reaching moisture soil. Since the root cap is not directly connected to the plant's vascular system, the water content of the root cap cells can provide a good indication of the moisture content in the soil. The root cap cells will produce the hormone abscisic acid as the soil dries. Production of abscisic acid stimulates root elongation by suppressing the synthesis of ethylene. The presence of ethylene is responsible for lower rates of elongation and an increase in diameter growth. So the inhibition of ethylene results in roots that elongate and penetrate deeper into the soil. Plants can also respond to wind. Plants exposed to wind produce stems that are shorter and wider than ones grown in more protective locations. It is the flexing of the stem back and forth in the wind that triggers an increase in the synthesis of ethylene, which in turn causes plant stems to expand more in diameter and less in length, leading to shorter and thicker stems. In the last lecture, we covered how day length can affect the timing of flowering. Day length also plays a role in preparing plants for overwintering. The shortening day length in autumn can trigger plants to reduce internode elongation, stop the production of photosynthetic leaves, and form storage organs. The reduced day length also causes some plants to prepare their meristems for winter. The plants begin to form bud scales to surround the meristem and to protect it from ice and water loss. In addition to the bud scales, there are a series of metabolic changes that allow meristems to remain in a dormant state during the winter. The plasmodesmata between the meristem cells and the other cells of the plant are blocked by plugs, which will be then removed in the spring. These plugs permit any growth-stimulating compounds from reaching the meristem. If you have any questions, please let me know via the Q&A discussion board.